So here we go. The phylogenetic tree. This is the star of the show. This is the learning that you're taking away from this lesson. The taxonomy and the morphology and the Carl Linnaeus and some of the background things about wardrobes and fashion, that's all preamble and review. The phylogenetic tree is what you're taking away from this week. And that's what the product of the week is also focused on. So, if we want to show the evolutionary change and how the organisms are related to each other, we normally do this in something known as a family tree. And so we've seen this when we talk about our own family. So we're going to use this for something larger in terms of classification. Once again, I'm going to come back to that book, The Tangled Tree. I really like this book because there's so much that is not told in the textbooks. There's so much that's not told in our typical classical lessons. Everything is, there's A that was discovered, and then there's B that was discovered. C relied on B's information, and A's information is all very linear. But science is riddled with many conflicts and many disagreements and many people being ridiculed and bullied, to be honest. I can use that word. They're bullied. And one of those people is this dude, scientist by the name of Carl Weiss. I'm not sure how to pronounce his name correctly. I should have looked it up. And Carl Weiss, he proposed, based on the evidence, he looked at the chloroplasts of plants, and he looked at the mitochondria of animals, and he noticed that these organelles had DNA that was different from the genomic cell, the host cell, the eukaryotic cell that's found in the nucleus. So that was odd. He also noticed that the DNA of the chloroplast and the DNA of the bacteria, so the DNA of the mitochondria, it looked different than the proposal of the endosymbiotic theory, which we talked about last week or the week before, was that some ancestral cell ingested a prokaryote, and then the two of them went through a symbiotic relationship, and one of them produced sugar from sunlight energy, and the other one used the sugar to produce work energy, ATP. But as he studied it, he recognized that the DNA inside these organelles was actually quite a bit different, and it got him thinking. And as he kept on studying a variety of very interesting prokaryotes, we'll call them, these single-celled organisms, he noticed that there was a difference. Most of the bacteria were here, but there was a group of bacteria that were not like them. And then he proposed that they should be put in their own domain because they're very different. The same way that an animal is different from a plant, these bacteria here were different from these archaeans back here. And so he proposed that there should be a new domain of life. Before this, there was the prokaryotes of bacteria and the eukaryotes, which is everybody else. And he said, well, it's in the prokaryotes, it's not just bacteria, this is somebody else. And for years, other scientists ridiculed him, laughed at him. They tried to ban him. They told people that were his colleagues, other scientists would say, you know what, you can't hang out with Carl. If you hang out with Carl, your career's done. Because everyone's going to think you're a laughingstock because he has no idea what he's talking about. He had a feud going on with this person, Lynn Margulies, and other people who, it just went on for years and years and years. And what's funny, as the scientific evidence came in and our technology got better and they're able to analyze the DNA, they found that the proposal that he put forward of the archaeans being a third domain was actually true. And about 20 years later, it became mainstream science. Everyone's like, oh, this is true, oh, this is true, oh, this is how it is. And then within 30 years, every textbook talks about the 3 domains of life. So it's an interesting story if you ever get a chance to read beyond what we do in the classroom. So if we just follow the history a bit, we'll see that in the 1700s, we had the animals and the vegetables, right? That was Linnaeus. In 1866, Ernst Haeckel, that person who did that amazing tree, but also said ontogeny recapitulates phylogeny. Protists, small, tiny little animals, but they're not, animals and plants. In 1925, another scientist proposed the prokaryotes, of which there was only one group, the monarans, and the eukaryotes, which included the protist animals and plants. In 1969, the prokaryotes still remained a group, and the eukaryotes now had fungi, because fungi were considered animals and plants depending on what kind of fungi you were looking at previously. And then 1977, Carl Weiss, that person I was talking about, he proposed bacteria as a domain, archaea as a domain, and eukarya as a domain. The 3 domains of life. That's what we have. That's the current hypothesis. Will this hold true? Well, time will tell. Probably not. We found some very large viruses that are the size, almost the size of bacteria, that have very complex genomes that we would not really classify with viruses. But they're not bacteria, so where do we put them? So the truth is, by the time another 5 years passes, there might be 4 domains of life, and I'll have to rejig this PowerPoint again. Right? So this is a hypothesis. Understand that phylogenetic trees and classification schemes are hypotheses. They are a prediction based on the evidence and supposition how organisms are related to one another. All right? So by definition, when you study inherited species characteristics and other historical evolutionary relationships, we put them all together in this big visual diagram known as a phylogenetic tree. And this study is known as phylogeny. Phylogeny is classification based on evolutionary history. So building the tree, how do we do this? That's the remainder of this lesson. We've already talked about some of the language. It is a tree. So there's a root, there are branches, there are nodes. The root is the ancestor. The extant or descendants are at the tips of the branches, and then these little nodes here, they also represent something. They represent a speciation event. We talked about that previously. A speciation event is where a branch occurred, something happened, some kind of reproductive isolating mechanisms caused these species to no longer come back together, right? And these branches happen all over the place. You should be able to read the tree. So looking at this tree that I have here, somewhere on here I can get my laser pointer. Oh, it's not coming up. All right. So somewhere along here, if I move my mouse here, what I'm moving on right here, this is a common history of A, B, and C. If I go all the way up here, that's a common history of B and C. If I go here, that's just C. This is just B, and this is just A. So you should be able to read the tree. It's pretty straightforward. It's not surprising how it'll come together. Now, if I want to go into a little bit more detail, any ancestor of C that's only on C's branch is a unique ancestor of C. If it's on B and C's branch in this node or anywhere below that, that is a common ancestor of B and C. And of course, we have a common ancestor at this node of A, B, and C. Clade is one of the terms I've asked you to define on your worksheet. A clade is a group of organisms that have a common ancestor. So you see a yes example here with the green. Oops, and you see a yes example here with the blue. This is not a clade because for this and this to have a common ancestor, you have to include this tip and you have to include this tip. This is not a clade because for this to be, then you have to include here. Right? For all these, you have to include this tip here. So you should be able to understand how a clade comes together. Cichlids, for those of you who have aquariums and you are into fish, cichlids are very popular. So they are pretty aggressive fish, and there's many different species. And it's very interesting because you found all these different species of cichlids all in one lake in this country in Africa. So do a quick search, look up cichlids, see what they are, and where they're found. I'm not going to pause. I'm going to keep on going, so you might want to pause the video. Cichlids are very interesting because they don't rely on a allopatric, they rely on sympatric. And so in the sympatric speciation, based on the fact they're all in the same lake, they found different parts of the lake to live in, and it's called a speciation event to occur. And so we can use this phylogenetic tree to show the relationship. And we see that these two here are closely related. You see that this one and these groups over here are not nearly as closely related. There's a much more distant ancestor. These classification schemes that we've used, they've been changing. So here's one from a long time ago. Well, clearly it's not something that's appropriate for us to use now, but you can see that these classification schemes have really evolved. In terms of phylogeny, the most common misconception that occurs is that if you're on the right or at the top or at the end, you're the most advanced. But phylogenetic trees don't show that. They don't talk about what's more advanced and what's more complex than the other. They just show how things are related and how closely their ancestors are, how close their ancestors are. So if I look at this diagram here, I know the pine and the rose are very closely related. I know the fern is also somewhat closely related. I know the moss is least closely related. That's what the phylogenetic tree tells us. So A, B, C, and D, it's not A is the simplest and D is the most complex or the advancement. It just tells us about relatedness. There are other diagrams you can use that can show complexity like this, but this is not one of them. Okay? We can infer that, but let's be clear, that's not what a phylogenetic tree is supposed to tell you. Okay? Now, the interesting about phylogenetic trees is that you can draw them in a variety of ways. So here's one way. This is the classic way, right? Pine in the rose here. But I can also do this. If I look at this phylogenetic tree on the right, the pine and the rose is still the two most closely related, with the fern being third and then the moss being fourth. Already talked about misconception with us. So if you were on the Google Jamboard and you made the phylogenetic tree for chimps and bonobos, you would have seen that the bonobos and chimpanzees are based, for you as a regular person, you couldn't tell the difference. Okay? You'd have to be a specialist to be able to tell the difference between a bonobo and a chimpanzee. Bonobos were discovered recently in the grand scheme of animal discovery, and when people first discovered them, they actually thought they were chimps, but they're not. So one of your tasks is to construct a tree. So in order for you to construct a tree, there are certain rules that you need to know. One of them is that when scientists want to construct a tree, they need to figure out what characters of the organisms are prevalent. And they use those prevalent characters in order to group the organisms into a particular clade or classification. So we have our classic animal kingdom phylogenetic tree, where the fish are in an area, the amphibians are in an area, the synapsids, which are the mammals, are in an area, the sauropsids with the reptiles and birds are in an area. And they have certain characters that allow us to classify them together. Those characters that we use to create phylogenetic trees have a specific name. They're called shared derived characters. So if I look at this particular tree here, I'll see that all these organisms have something in common, and that is that they have a backbone, right? It's not listed on here. And then as I go further up this tree, sharks, they branch off because there's something that ray-finned bony fish have that sharks don't. And it might be the air bladder or the gills or some other feature, but something the ray-finned fish have along with all of these animals here. 4 limbs is a shared derived characteristic of amphibians, turtles, lizards, snakes, crocodiles, birds, and mammals. They all share it, so they're all grouped together. And as we keep on going up, we're going to find other things that will allow us to classify certain organisms more closely to each other. All right? So in this next activity, you'll see Milkshake here and Pebbles and Tsunami. And I just took these 2 pictures. Actually, I took this one today, earlier today. It was a beautiful day. And I took this picture a couple of days ago. And so in this activity, I quickly want you to spend a little bit of time classifying them. What characteristics are you going to look for that are going to allow you to classify, clearly these 2 are somewhat related, but what about them are you taking out to relate them in the same category? And we know that this Milkshake, this turtle, and this tortoise are related. They're all reptiles. What about them allows you to classify them into the same category? What allows you to classify turtles, tortoises and turtles, the same thing pretty much, in the same family? So there is certain characteristics. So go ahead, take a moment. I'm going to move on, but you want to spend some time thinking about this so that way the learning is more meaningful. All right, so we're ready to classify. And understand when one person does it, it might be slightly different from another person. Linnaeus used this system. Domain, well, he didn't use domain, but domain, kingdom, phylum. Does King Philip come over from Germany swimming? Right, so there's this. Phylogenetic uses evolutionary history, but the 2 of them match up. Typically, phylogenetic classification will corroborate the Linnaean taxonomy. The 2 of them go hand in hand. It's not very common when the 2 of them are in conflict. But when they are in conflict, we'll typically go with the phylogenetic classification over the Linnaean because the evolutionary history is supported by the bone structure, the homology, it's supported by the DNA, and so on and so on and so on. Okay? So the phylogenetic system is definitely superior. When you're ready to create these, we rely on a concept known as parsimony. It's a great concept. In language, we use a thing called Occam's razor. You'll see it in a lot of books. And often Occam's razor and parsimony have the same kind of definition in that the simplest answer is typically the best answer. So when we start looking at different ways that organisms can evolve, we're looking for the system that involves the fewest steps. So if one evolution pathway takes 5 steps, another evolution pathway takes 10 steps, typically the 5-step pathway is more accurate. Because evolutionary change that is beneficial and leads to new species is extremely rare. It seems so common when you look at the planet, you see millions of different organisms, but the actual event is very rare. So parsimony is an important concept. The simplest answer is true. So here is a table. This is a shared derived characteristic table. So vertebrate, having a backbone, all these categories of animals have a backbone, yes. Bony skeleton, well, the shark doesn't, so that's a no. And so if we start grouping them, all of a sudden the shark, because it has this one missing feature characteristic, it's an outgroup. 4 limbs, well, the bony fish don't have 4 limbs, and the other ones do, so they're all going to be grouped together, and that's a no. Amniotic egg is a special egg. The chicken egg that you guys have, that's an amniotic egg. The fish eggs that you might see, those are not. So an amniotic egg has a yolk and some other several features. Hair. The primates and the rodents and the rabbits all have hair, so we might group them together. The special eye bone structure, we see that with crocodiles and dinosaurs and birds. So we group them together. So this shared derived characteristic table, already I can see some commonalities. All right? So one of the things I'm going to ask you to do before you go on to the next slide is I want you to circle anything that looks like it could be a clade, right? Anything that looks like it can be a clade, circle it, because that's what you're going to use to build the phylogenetic tree. So you might want to pause it here while you work through that in Nearpod, because you can circle that in Nearpod if you're following along, and then I'm just going to keep on going. So here is the various clades circled. There's a big one that includes all the vertebrates and all the bony skeleton vertebrates and all the 4-limbed tetrapods and all the amniotic eggs and so on and so on. And then from this clade, you can create a phylogenetic tree where you have the ancestral vertebrate and everybody's included, and then the outgroup of the sharks, and everybody's included under the bony skeleton except for the sharks. And then the 4 limbs, everybody's included except for the bony fish, and so on and so on. And you'll see that the primates and rodents and rabbits are grouped together, and the birds and reptiles are grouped together. And that's how we create a phylogenetic tree from a shared derived characteristic table. Now, once again, I want to emphasize this. So on the Nearpod, you're going to see a slide here. So you're going to pause again on the video lesson, and you're going to go to the next slide, and I ask you, from these 20 organisms, how many different possible combinations can we have? So you've paused me, you've made a prediction, you've typed it in, all right? And the truth is, with 20 different species alone, remember we have millions, there are, what is that number? Let's see, we got hundreds, thousands, millions, billions, trillion, quadrillion, quintillion, sextillion. So 8 sextillion possible combinations. That's a lot. So how many of those are actually true though? 1. But it could change based on new evidence. So even though only 1 is right, it could change as we get more evidence, and you might go on to a different possibility. So that is the phylogenetic tree. So you'll notice in the lesson I've asked you to come up with your own phylogenetic tree before you looked at this slide, so I might have jumped the gun by clicking on this, but anyway, hopefully you saw my Nearpod and you saw my asking you to make up your own proposal, but that's a purple, so you just keep on going through the lesson if you want to get to the real activity. So if we were in class, at this point we would have brought in a whole bunch of Oreo cookies and Voortman cookies and Dad's cookies and cookies, and we'd all be eating cookies. Before we ate our cookies though, you would create a phylogenetic tree. So you get to do this here. So in the Google Doc, you're going to see this section that has the cookies. And these cookies are movable, right? So you can move them around the page. And what you're going to do is you're going to create a phylogenetic tree. This cookie here is your common ancestor. So for many of you, you might not be as comfortable working with Google Doc. You might be more comfortable working on slides. You can take these pictures and put them on slides or PowerPoint, and you're going to create a phylogenetic tree. And you're going to show me the ancestral cookie, and then you're going to show me how the lines break out. You're looking at the features, right? This cookie has has wafer and chocolate. Wafer and chocolate, that's rectangular, that's circular, that's rectangular, that's circular with chocolate lines, and that's got chocolate all over the place in this one. So how does it come together? There's many different answers. So work by yourself, work with a partner. I don't care who you do it with. You're going to work with this, and then you're going to post your answer up on the Flipgrid. And then I'm going to critique your answer. And when you post your answer up, you're going to tell me why you put them in that particular format. Okay, and so there's many different answers. I'm going to give you an answer immediately. So as soon as you get to this, post it up on the Flipgrid. And then you'll see on the Nearpod, there's a link for the Flipgrid. There's also a link for the Flipgrid in the Google Classroom. Just do it. I'll give you feedback immediately, and that will prepare you for the actual activity. For those of you doing the minimum, then you can stop here. For those of you who actually want a complete understanding, you're going to work through the phylogeny activity. So the phylogeny activity, I've put it into the PowerPoint so you can see. It's a mapping activity. So once you've made it to this spot, you'll notice at the end of the Nearpod, there's an exit. It tells you what you should have learned, and there's an exit. This is the kind of question, if we were doing a test situation, this would be on the test. I'd ask you a question like this on the test. So bring us to the end here. You have this CK12 digital textbook, so that's going to go through phylogeny cladistics, so it's an online textbook. So you click on Start Practice after you've read a couple pages here and watched this person's video here. You can start the practice. And a Khan Academy video as well. It's like 10 minutes long. These are, you don't have to watch them. You can skim through them if you want more learning. The Learn 360 resource as well. It's really connecting to taxonomy and through different styles of classification schemes up until we get to phylogeny. And then the phylogeny activity. So I'm going to give a very specific video lesson set of instructions on how to work this activity. To give you the Coles Notes version, so now that we all know what Coles Notes are because I've explained it, you're going to take these organisms. So these are fake organisms. You'll recognize them as other things. You take these organisms and you need to take at least 10 of them for complete, all of them for exemplary. And you're going to create a phylogenetic tree from these organisms. You have to figure out which one of these is going to be your ancestral organism. If you go into the D2L on the calendar, I'll give you a link to the phylogenetic exercise. So right now with these, all you can see is one view. But if you go into our course in D2L, so D2L grade 11 biology, and I'll put a link up on the calendar and it'll be easy to find. You see this little piece of paper? It's the same piece of paper that's in each of these. So see how the piece of paper here looks big and here it looks small? That tells you that this particular object is huge compared to this. I didn't do it in all the, but I did it in many. So I'll give you a relative size for these. So these are your organisms. And I know you're going to be tempted to call it a spoon and an egg separator and a turkey baster and a knife and a whisk and all that kind of stuff. But they're organisms. So you're creating a phylogenetic tree. So you can take these and you can move them around. You can, once again, use PowerPoint or slides or move them around here. If you're using a Word document, you're going to have to change the format so it can be moved. Otherwise, it'll be stuck on the page. Right? And under evolution, this phylogeny. So you want to see a better view of these. Under today's date and for the rest of the week, there'll be a thing on here. It's called, where is it? Lesson 7 Phylogeny. And you'll see phylogeny activity, photos, examples. So if I click on this, it's a web page. And on here, ignore all these instructions, but if you click on any one of these graphics, you'll get a bigger version. Right? So if you're trying to get a little bit more detail of what this looks like, because you can't see it very well, click on it and you'll get more detail. And on this same site, you'll see 3 versions of every single thing. So the knife, you'll see from this, or you can see from a different angle, right? So this just gives you more detail because you can't actually handle them in your hands. Right? And there's that little piece of paper, right? So you're looking at the hammer, you can clearly see it's got 2 little segments or 2 little branches here. That's something that you might use to help you classify your organisms. So that's in D2L. So those are your organisms. What you're going to do with those organisms is create a phylogenetic tree. That's part 3. See, it's in yellow here. Create a phylogenetic tree or cladogram. If you complete this activity, that's what you must do. So you can construct it. You can just take a look at it and see, oh, these kind of look similar. These kind of look similar. I'm going to create my phylogenetic tree, and then you must explain it. In your analysis, you must tell me 3 types of information that you can take from the cladogram. You must tell me how many speciation events you think occur. You must pick any 3 branches and tell me that the knife ended up getting more serrated edges that allowed it to better dig into the ground, and it ended up becoming a screw shape. Or the screw went from being small to being larger so that it could, go more quickly into the ground or something like that. Identify on your phylogenetic tree a convergent evolution. So do you understand the concept? Speciation event, extinct, extant, micro, and macro evolution. So I'm looking for all those things on your tree. You're going to find any 2 other organisms that you deem appropriate. Is your tree representative of gradualism or punctuated equilibrium? This question here, genetic level, so genetic diversity. Your rebop, if you remember working on that once upon a time, it could be connected to your tree, so how would you do it? Okay. So these are the analysis questions of the tree that you create. There is a part 1 and a part 2 of this. And the part 1 and part 2, you'll notice it's in purple. So one of them is a shared derived characteristics table, and the other one's a characteristics table. So you're kind of going through each one and you're writing down observations that you notice. The spoon. I know it's a spun, but work with me, it means spoon. And I think there might be a couple that are missing on there. Just add them if you want. Okay. So that's just an observation table. And the next one is a shared derived characteristics table that you can use, and you can put X's on all the things that are shared. So this has serrations, this has whorls, this has silver, this has plastic, whatever you want. And so that might help you group it. But those could take a long time with this many organisms to do. And so some of you might find it easier just to go and jump right into the creation of the phylogenetic tree. That phylogenetic tree can be worked on by yourself, with a partner, with a group of 3, with a group of 4. Just tell me who's working with who and try to make sure that everyone's working on it together. Okay? And you'll notice that's in yellow. So that's your activity. That is the lesson. I will make a secondary video lesson, and I'll show myself manipulating these pictures and creating a phylogenetic tree from the pictures. Of course, you can't use the one I create, and hopefully that'll help you have more success with this particular endeavor. All right, have a good week, and I will talk to you soon.